The overall goal of this procedure is to extract quantitative information on plant roots from in-soil X-ray computed tomography data.
This method of investigating the root structure of live plants can help answer key questions in the biology field, such as how plants respond to changing environmental conditions, like drought, microbial activities, and so forth. The main advantage of this technique is that it is non-invasive. Therefore, it allows us to study plants in their native environment, the soil.
Visual demonstration of this method is valuable as the procedure involves numerous steps, including tomographic imaging, data reconstruction, and analysis, which may be obscure without visual aid.
To begin, place a potted plant on the sample manipulator of the X-ray tomography scanner at a distance desired for target magnification. For a plant in a 2-inch diameter holder, set a sample to a source distance of about 3 inches. In the instrument control software, set the X-ray power settings to 85 kilovolts and 190 microamps. Then, set the exposure time to 1 second for a better signal-to-noise ratio. Set the number of projections to 3,142 and the number of frames per projection to 4 for good data statistics. Using the scan parameters just set, select the Shading Correction tab, then click on New and name the shading correction file. Then click Present to confirm the new conditions. Next, click Create and click OK to using 2 reference images. Then click Generate. Then, when the Warning pop-up window appears, physically remove the sample from the beam path before clicking